Welcome back. We're into chapter 6, the tour of the cell. This chapter focuses on the components of cells and what they look like and how they function and how they interact together. The bulk of the chapter really focuses on cell structures and their functions. There are other components of the chapter that we want to look at as well. The first concept in chapter 6 is on microscopy, and just read that over. Get a feeling for the different kinds of techniques that we use to study cells. And then we'll move into, in this video, cell types and size limitations. So, when we talk about cells, we use the terms prokaryote and eukaryote a lot. What are the differences? How do we know what type of cell we're looking at? Let's take a look at some questions. And as we answer these questions, we'll be able to sort out the differences between prokaryotes and eukaryotes, okay? So let's start. These are kind of questions for you. Maybe you know the answers to these already, maybe not. Whoops. We'll see. Okay, first question. These are tiny cells, about 1 to 10 micrometers in size. Now, just for a reminder, there are 1,000 micrometers in a millimeter. So these are very, very small cells. Which cells are smaller? Turns out the prokaryote cells are smaller. These prokaryote cells are 1 to 10 micrometers on average. Eukaryote cells are cells that range on average from 10 to 100 micrometers. Oops, that's not written well. That is micrometers. Much larger on average. All right, how about number 2? Endoplasmic reticulum is an organelle found in these cells. Endoplasmic reticulum. You might not remember what that does, but it's a membrane-bound organelle. Eukaryotes have membrane-bound organelles. Prokaryotes don't. In fact, that's kind of why they have their titles. The word prokaryote means before, karya, kernel, before a kernel, before the membrane-bound nucleus. Prokaryotes have no membrane-bound organelles. Eukaryotes have membrane-bound organelles, including a nucleus, endoplasmic reticulum, Golgi, mitochondria, chloroplasts, lysosomes, you name it. All right? So when we think about membrane-bound organelles, let's just remember that. That refers to cells that are eukaryote. Okay? Part 3 goes with this. Question 3 says, the nucleus in these cells has 2 membranes. Well, the nucleus is found in a eukaryote. The word eu means true. Karya is true kernel. The nucleus is the true kernel of eukaryotic cells. And so the eukaryotic cells have nuclei with membranes around them. Prokaryotes have nuclear regions but no membranes around their genetic material. All right, well, what are some of the prokaryotes and eukaryotes? Let's get to questions 4 and 5. E. coli is this type of cell. E. coli is a bacteria. You hear about it usually negatively, but we have E. coli living in our guts, doing all sorts of things for us all the time. But E. coli are bacteria. Bacteria are prokaryotes. There are 2 major categories, major domains of prokaryotic cells. The bacteria, domain bacteria, and domain archaea, which also includes a group, this is AEA here. Wow, that was a mess up. Archaea and bacteria include cells that are typically single-celled and simple. the prokaryote cells, small cells without membrane-bound organelles. Eukaryotes would include all the rest of the organisms out there. So fungi are eukaryotes. Fungi, animals, what else? Plants. These are all eukaryotes. And what we used to call protists, mostly single-celled organisms, we called them protists. Things like algae and euglena and amoebas and things like that. Those were all eukaryotes. They are more complex. They have the membrane-bound nucleus and membrane-bound organelles. Number 6, DNA is the genetic information in these cells. So what is the heritable genetic information? Well, we have DNA, so eukaryotes have DNA. So do all plants, animals, fungi, protists have DNA as their genetic material, but so do prokaryotes. DNA is the heritable information in all living things. Prokaryotes and eukaryotes. So that was kind of a bad question, right? Or a trick question. Number 7, ribosomes are found in these cells. Now, ribosomes are tiny little structures that you probably learned are the site of protein synthesis. And so you would think, oh, I bet they're in eukaryotes. They are. They help make proteins in eukaryotes. Are they in prokaryotes too? Yeah. Okay, ribosomes are found in prokaryotes and eukaryotes. They are the site of protein synthesis. They help make proteins in both of these types of cells. All cells need proteins to do everything that they do. So that's kind of a trick question, number 2. Question 8. The genetic material in these cells is composed of multiple linear strings of genetic material called chromatin. All right. Eukaryotes have, if we draw a eukaryotic cell, it has a nucleus, and in its nucleus are strings of DNA. Those strings of DNA are linear. There are lots of them. In humans, we have 46, and we call that chromatin. It coils up sometimes, and we call those chromosomes. Prokaryotes have a nuclear region, and in it they have their DNA, but their DNA is circular. It's still double-stranded, but it's circular. One circular piece of DNA. So they do not have multiple linear strings. Those are the main differences between prokaryotes and eukaryotes. Here are some slides of prokaryotes and eukaryotes. This one represents kind of some of the basic things found in bacteria or archaea. This happens to be a bacterium. And you can see it has a single chromosome or a single circular DNA material. Okay, and it doesn't have membrane-bound organelles. Here's an animal eukaryote. So our animal eukaryote has the membrane around it. There's the nucleus. And you can see all sorts of other structurally different organelles that we're going to take a look at later. Here's an example of a plant cell. It's a little bit different than an animal cell. It has a few different things, including, for example, chloroplasts. All right. Why are cells so small? So when we think about cells, we realize that most cells fall in this category, somewhere between 1 and 100 micrometers in size. That's pretty small. And even cells in bigger animals or bigger plants are still that small. They don't grow big. Why not? Why are cells so small? We think it has to do with their jobs. Okay? In order to carry out metabolic activities, it's better for cells to stay small. Because small cells, well, just think about cells for a minute. cells have a lot going on inside them. But they need to transport materials into the cell and out of the cell, right? Some, oxygen and nutrients have to come in, chemical signalers, etc., and wastes have to go out, maybe hormones that the cell is sending out, or other kinds of enzymes. And if the cell grows too large, the volume grows faster than the surface area. And that's what this is kind of showing you here. You can check it out. As the cell grows, its volume grows faster than its surface area. That's bad for a cell. It won't be able to maintain all of its functionality with big volume with small surface area. It needs to be very small instead, so that surface area relative to the volume can stay large. And that's why we think cells stay very small. That's it for now. We'll come back to take a look at cell parts.